Fire, humanity's oldest foe. Left unchecked, it consumes all in its path, leaving only death. And yet, fire can also be beautiful, majestic, utilized by humankind. By wiping the ground clear, fire can make way for new life. In fact, burnt soil is often far more fertile and allows for faster plant growth in the aftermath of fire. But what makes this the case? Not everything dies in a fire. Within the scorched soil, invisible to the naked eye, millions of tiny organisms continue to grow. With the onset of climate change and severe drought conditions across sunny California, the frequency of wildfires is expected to increase drastically. The potential effects of this mass disturbance on soil bacteria, on the ecosystem, and on humanity lay shrouded in mystery. Enter the scientist. Intelligent and highly motivated, the undergraduate researchers at UCLA devote countless hours each week to studying bacteria collected from the soil affected by the recent Skirball fire in Southern California.
We know that plants tend to grow faster in soil after a fire. So our study is to determine whether the bacteria in that soil play a crucial role in promoting plant growth.
Bacteria are commonly associated with plague and disease, but this is a gross oversimplification. In fact, bacteria are responsible for a number of processes essential to both of the environment and the human body, such as fertilization and suppression of disease.
Bacteria also take nutrients and make them more usable and available for plants to grow. One of these nutrients is phosphate, and it's normally not very present in the soils, and so plants are very limited by it. So what bacteria can do is solubilize it and make it usable. Another thing they can do is protect plants from disease by outcompeting with pathogenic bacteria and then making a protective area around the plant roots.
Measuring this bacterial activity is no simple task. Soil is carefully collected and sieved to its smallest components. Then it is dissolved in water and placed onto agar plates, containing the nutrients to allow any soil bacteria to grow. Only once have the bacteria started to grow in a lab setting can the actual experiment begin.
So one of the things we wanted to measure was the ability of our bacteria to solubilize phosphate and turn it into a usable form. So we grew them on these purple plates here that have a form of phosphate that they normally can't use. And if they turn it into a usable form, they'll have an acid byproduct that changes the pH and the color of the plate. So you can see around this growth here, there's actually an orange kind of color change on the plate that signifies that this isolate RO6 changed the phosphate.
To outcompete pathogens, soil bacteria have developed ways to produce and resist substances that kill undamaged cells, commonly known as antibiotics.
A lot of us might not know this, but we owe the production of antibiotics to soil microorganisms like bacteria and fungus. And by studying the effects of antibiotic production and resistance in these microbes, we can learn more about the effect of antibiotics in plant protection, fighting human disease, and if we're lucky, we might even find new compounds or chemicals to help us fight disease in humans.
So in order for us to determine whether our soil bacteria candidates produce antibiotics, we first grow them together in a plate with a common bacteria. And so if they are producing antibiotics, they should be having a clearing such as this.
With any luck, their data can be useful in comparing the bacteria in the two samples. However, their work is still not done.
So this is the MIC daptomycin test. We use the daptomycin script that we place on the middle of the plate. So it is with a lot of the isolates that we use for antibiotics resistant assay. And the numbers shows on the script is the concentrations of daptomycins that the script is secreting. So we can see how much daptomycin antibiotics that the isolates tolerate.
So to determine the level of antibiotic resistance in our soil bacteria, we grew them on plates in the presence of antibiotics, which are actually on these little discs. And if they are resistant, they will grow unaffected by the drugs. But if they are sensitive, then they will show zones of growth inhibition like this. And if they mutate and become resistant to those antibiotics, then they'll show mutant colonies like this in those zones of growth inhibition.
For the undergraduate researcher, in its natural habitat, the study of these soil bacteria is full of setbacks and obstacles. However, they have evolved to tolerate these harsh and frustrating conditions.
You do everything 3 times, and I'm not sure it's working.
Despite these obstacles, the undergraduates eventually succeed in meeting their deadlines and completing their experiments.
So from our experiments, we actually determined that the amount of phosphorus in the burn soil was significantly higher than in the control soil. So that suggests that maybe the increased plant growth in the burn soil is due to a higher amount of usable phosphate, which could be coming from these different bacteria.
So in addition to these tests, we're going to take the DNA from our bacteria and sequence it. And by doing this, we can actually identify what kind of microorganisms live in our soil samples, and we can look at changes in microbial composition after a fire.
DNA sequencing has progressed rapidly in the past decade, and now can be done quickly with minimal problems.
This is an ongoing project with a lot more to discover, and we hope that the work that we do here at UCLA is beneficial for future projects and researchers all around the world.
With the changing climate and environment, much of our future is still left uncertain. However, as the Earth continues to transform, we must turn our attention to what lives in the ground beneath us, and hopefully harness it for the benefit of society.